Howdy, this is Lab Report 3, Staining Lab. Simple, gram, acid fast, capsule, and endospore. Group 2, Ana Martinez and
And Hannah Garcia, Microbiology Fall 2024.
Introduction. The purpose of this lab is to learn how to perform different staining techniques on different types of bacteria and be able to examine their characteristics under the microscope. This lab is crucial to understanding why different staining techniques are used in microbiology. Significance and background information. Performing different staining techniques is crucial to differentiate bacterial types based on their cell wall properties, morphology, and arrangement. These staining techniques can reveal specific cellular structures such as endospores, capsules, flagella, etc. Staining helps visualize bacterial populations and characteristics, growth patterns, and host cell interactions. Ultimately, staining techniques are significant for diagnosing infections and determining the appropriate treatment by understanding the biology and virulence of different bacteria. The method for preparation for broth. Get a clean slide and label it with a wax pencil and make one or two circles. Gently swirl the tube with the bacteria. Sterilize the inoculating loop, open the tube, flame the top of the tube, dip the inoculating loop in the tube and take it out. Then flame the top of the tube before closing. Transfer the drop of culture broth to the circle on the slide. Repeat in order to transfer enough bacteria. Spread the drops to the size of a nickel. Place the slide on the warming block to air dry. For gram stain, heat fix by passing the slide over the flame about 6 to 9 inches over the butane can 3 times. The materials and methods. These are the materials. Place the heat fix slides on staining tray and flood slides with crystal violet stain for 1 minute. Then rinse with deionized water for 5 seconds. Next, flood slides with grams iodine mordant for 2 minutes. Then rinse with deionized water for 5 seconds. Decolorize slides with acetone and immediately rinse with water for 5 seconds. Flood slides with counterstain, safranin for 1 minute. Then rinse with deionized water. Blood dried slides with bibulous paper and examine under microscope. These are the materials. Place the prepared slide of Mycobacterium sp on the staining tray and flood with quinone carbonyl fusion stain for 2 minutes. Then rinse slide with deionized water. Rinse slide with acid alcohol decolorizer until no more color drains from the slide for about 3 to 5 seconds. Then rinse slide with deionized water and shake off excess water. Flood slide with counterstain, methylene blue or brilliant green for 30 seconds. Then rinse with deionized water, blood dry with bibulous paper and examine under microscope. These are the materials. Pour a small drop of crystal violet on the slide. Add a loopful of Klebsiella pneumoniae to the slide by doing the aseptic technique. Mix it into the dye, but do not spread it out. Use the second slide to drag the stain cell mixture into a thin film along the slide and let it sit for 5 to 7 minutes. Then allow the slide to air dry. Do not heat fix. Examine under microscope and look for unstained capsules. These are the materials. Prepare a heat fix smear of Bacillus subtilis and place it on the staining tray. Cover the smear with filter paper and saturate it with mal- malachite green stain solution. Use a clothespin to place it over steaming water and do not let the slide dry out. Leave it for 5 to 7 minutes. Remove the slide and let it cool. And rinse with deionized water over the staining tray for 30 seconds. Counter stain with safranin for 1 minute. Then rinse with deionized water for 5 seconds. Blood dry with bibulous paper and examine under microscope.
Results and observations. The following slides are the result and are observations of different bacteria and performing different staining techniques. The pictures are our results, and the observations are based on what we recorded on our lab notebooks. Gram staining. Staphylococcus aureus is a gram-positive bacteria with a coccus shape and Staphylococcus arrangement in clusters. For Bacillus cereus, the cells are bacillus or rod-shaped in Streptobacillus chains, and it is gram-positive. Salmonella typhimurium gram-negative bacteria, bacillus rod-shaped with single rods. The mixed culture consists of both gram-positive coccus or Staphylococcus aureus and gram-negative bacillus or Escherichia coli. For our unknowns, 102 is a gram-positive bacteria with rod-shaped cells and Streptobacillus arrangement. 202 is a gram-negative bacteria with a coccus shape and Staphylococcus arrangement, and some observable purple stains as well. Acid-fast staining. Mycobacterium was both positive and negative. The positive had a pink color and was bacillus or rod-shaped, and so was the negative Mycobacterium, only it was blue and purple in color. Klebsiella pneumoniae for the capsule staining included cells with a bright capsule or halo-like appearance around them. And for endospore staining, Bacillus subtilis had terminal endospores that were green and mature, surrounded by red vegetative cells. In conclusion, microscopy and staining techniques are important when in the fields of clinical microbiology, environmental microbiology, pharmaceutical research, among others. The microscope allows us to see a detailed observation of microorganisms for us to study based on their shapes, sizes, and arrangements. Different staining techniques add contrast to enhance bacterial characteristics under the microscope. Overall, these techniques are vital to increasing our understanding of microbiology, improve diagnostics and treatments, and advance in microbial research. Thank you.